Introduction to Medical Microbiology. Medical microbiology is the study of microorganisms that cause diseases in humans, the ways these organisms interact with our bodies, and the methods used to detect, prevent, and treat the diseases they cause. It bridges biology, medicine, and laboratory science. The field deals with 4 major groups of pathogens: bacteria, viruses, fungi, parasites. It also involves understanding our microbiota, the beneficial microorganisms that inhabit our body, and how they interact with pathogens. In a clinical context, medical microbiology serves 2 main purposes: diagnosis and treatment of infectious diseases, prevention and control of infections in healthcare and community settings. 2. Microorganisms in the biological system. Microorganisms are the smallest forms of life, yet they play enormous roles in ecosystems and in health. In the biological system, beneficial roles include nutrient cycling, aiding digestion, producing vitamins, and protecting against harmful microbes. Harmful roles involve causing disease, spoiling food, or producing toxins. The major categories are prokaryotes, bacteria and archaea, simple cell structure, no membrane-bound nucleus. Eukaryotes, fungi, protozoa, and certain parasites, more complex cells with membrane-bound organelles. Viruses, acellular entities that require host cells to reproduce. 3. General and special bacteriology. General bacteriology covers the basic structure, physiology, and classification of bacteria. Bacterial cell structure, cell wall, plasma membrane, cytoplasm, ribosomes, nucleoid, plasmids, flagella, pili, and capsule. Staining techniques: gram stain, acid-fast stain, spore stain. Growth requirements: temperature, pH, oxygen levels, and nutrients. Growth phases: lag, log, stationary, death. Special bacteriology focuses on specific groups and their clinical significance, such as gram-positive cocci, e.g., Staphylococcus, Streptococcus. Gram-negative rods, e.g., Escherichia coli, Klebsiella, Pseudomonas. Acid-fast bacteria, Mycobacterium tuberculosis. Anaerobes, Clostridium difficile, Bacteroides fragilis. 4. General and special virology. Virology is the study of viruses, tiny infectious agents that cannot replicate outside living cells. General virology covers virus structure, capsid, envelope, nucleic acid. Modes of transmission: respiratory droplets, blood, sexual contact, vectors. Replication cycle: attachment, entry, uncoating, replication, assembly, release. Host-virus interactions: latency, persistence, immune evasion. Special virology examines medically important viruses, such as influenza virus, human immunodeficiency virus, HIV, hepatitis viruses, E. SARS-CoV-2, COVID-19. Herpes viruses, HSV, VZV, EBV. 5. Mycology. Mycology is the study of fungi, including yeasts, molds, and dimorphic fungi. Yeast, single-celled, reproduce by budding, Candida albicans. Molds, multicellular, filamentous, Aspergillus, Penicillium. Dimorphic fungi, grow as yeast at body temperature and as mold at lower temperatures, Histoplasma capsulatum. Fungal infections, mycoses, range from superficial, ringworm, to systemic, cryptococcosis. Opportunistic infections are common in immunocompromised patients. 6. Parasitology. Parasitology deals with protozoa, helminths, and ectoparasites that infect humans. Protozoa, unicellular eukaryotes, Plasmodium spp, Giardia lamblia. Helminths, multicellular worms, Ascaris lumbricoides, tapeworms. Ectoparasites, live on the body surface, lice, mites. Transmission routes include ingestion of contaminated food/water, insect vectors, and direct skin penetration. 7. Laboratory methodology in medical microbiology. Laboratory work in medical microbiology involves specimen collection, transport, processing, and analysis. Microscopy, light microscopy, electron microscopy. Culture, using selective and differential media. Biochemical test, catalase, oxidase, urease, fermentation test. Molecular methods, PCR, sequencing. Serology, detecting antibodies or antigens. Good laboratory practice requires strict adherence to biosafety protocols. 8. Bacterial physiology. Bacterial physiology studies how bacteria grow, reproduce, and survive. Metabolism, catabolism, breaking down molecules, and anabolism, building up molecules. Energy generation, aerobic respiration, anaerobic respiration, fermentation. Adaptations, sporulation, biofilm formation. 9. Pathogenicity and virulence factors. Pathogenicity is the ability of a microbe to cause disease. Virulence factors are the traits that enhance this ability. Examples of virulence factors. Adhesins help bacteria stick to host cells. Capsules protect against phagocytosis. Toxins, exotoxins, endotoxins. Enzymes, coagulase, hyaluronidase. 10. Bacterial genetics. Bacterial genetics examines how bacteria inherit and transfer genetic information. Mutation, changes in DNA sequence. Gene transfer, transformation, transduction, conjugation. Plasmids, extrachromosomal DNA that can carry resistance genes. 11. Antibiotics and chemotherapeutics. Antibiotics, naturally derived or synthetic compounds that kill/inhibit microbes. Mode of action, cell wall synthesis inhibition, penicillins, protein synthesis inhibition, tetracyclines, nucleic acid synthesis inhibition, quinolones. Resistance mechanisms, drug inactivating enzymes, target modification, efflux pumps. Chemotherapeutics also include antiviral, antifungal, and antiparasitic drugs. 12. Microorganisms and disease. Infectious diseases range from acute to chronic and may be localized or systemic. Examples: tuberculosis, malaria, influenza, HIV, AIDS. Understanding disease epidemiology helps in control and prevention. 13. Current research trends in medical microbiology. Research areas include antimicrobial resistance surveillance, vaccine development, microbiome studies, novel diagnostic tools. 14. Quality assurance, accreditation, and organization in medical microbiology. Clinical laboratories follow strict guidelines for method validation, quality control, and accreditation, ISO standards. Areas of responsibility include accurate reporting, biosafety compliance, continuous staff training. 15. Library science and statistics in microbiology. Researchers use library resources to find literature and apply statistics to interpret results. Descriptive statistics, mean, median, mode. Inferential statistics, hypothesis testing, confidence intervals.